Okay, so the first thing we're going to do is just load in this count matrix that we have. So that comes from this example data file that you should have downloaded. Um, so we're just going to read this as a pandas data frame, and it'll look um, in this familiar form of the genes as rows and the samples as the columns. So we have 6 samples here. Okay, so hopefully everyone has that count matrix loaded. And then the next file we'll also load is the metadata for each of these samples. So that will typically look something like this, or our row index identifiers are these GSM numbers, and then we have information about each of these samples in the columns. So your metadata table could have any number of variables included in here. Just for this example, we would just have the cell line and the cell type, which happen to correspond to each other. So our condition of interest that we're going to be computing the differential expression across, or we're going to do that across the cell lines. So I'm just going to set this condition variable equal to the name of that column, cell line. Okay, and then so before we begin the actual differential gene expression analysis, I'm first just going to do some of those pre-processing steps or the filtering steps that I mentioned in the lecture. So we want to remove any of these genes that are very unlikely to be differentially expressed. So genes with 0 or few read counts. And then you'll see later when we go to actually implement the DGE methods that some packages like DESeq2 already they have options for doing this inside their own functions. But it's totally fine if you do this yourself separately. So the low expression threshold, this is something you'd probably choose based on looking at your data. Just for the purposes of time, I'm just going to use this arbitrary number here. But yeah, to kind of make a good decision about this, you might want to first just plot a distribution of your read counts. And then I think DESeq uses some of their own calculation or some default value for filtering this. But then to filter out those genes with low counts, we're just going to apply this data frame filtering. And you can see, so we started out with 35,000 or so genes. And after this gene filtering step, we've removed a fair number of them. And so this filtering left us with about 21,000 genes. So you can definitely scale this filtering depending on how many genes you still want to be testing for differential expression. And then another check you can do is to look over the library sizes for each sample. So this is a way you could identify possible outlying samples, or then and look at the overall data quality. So any differences you see here in sequencing depth, these are going to be due to experimental variations or random technical noise. And if it's too outlying, then you should consider removing that sample. But normalization should hopefully address most of those differences. So library sizes are going to be these large read count numbers that we get. And yes, Notion, yeah, please do look at the video later once you have that running. Okay, so as long as we have our filtered or count matrix, it can be filtered. It doesn't have to necessarily be filtered. But now we're going to move on to actually implementing one of these differential expression methods. So here we're going to start off with DESeq2, and DESeq2, it's originally implemented in Python, same with Lima, or in R, excuse me, same with Lima. But they now have this PyDESeq2 library, which is a re-implementation of the same algorithm, but in Python. So we're going to import the methods that we need from DESeq2. So yeah, so like we talked about in lecture, we're going to give DEC2 the raw gene counts, and then the algorithm will do its own normalization. Um, so what that looks like is, so we first instantiate this inference object, and then what DEC2 needs from us is the count matrix, the metadata table. So that's going to tell uh, the algorithm which samples are in which group. And then you're going to specify the condition that you're trying to calculate the differential expression over in this design factors attribute. So if you have more than one condition that you're trying to test differential expression for, then you would provide a list of column names here instead of just one name. So I would maybe say like cell type and tissue type and whatever other variables you're trying to test. And then, so by default in this method, outliers detected with Cook's distance, which is like an outlier metric, those outliers are refitted into the model. So if you want to skip that, you can just turn off that parameter. And then we give it the inference object that we're going to get our results from. So we run this. So now inside that DEC data set object, we can call the method. So this will take a second, depending on how large your data set is, how many conditions you're testing. And it will do this. So for us, this was pretty quick. Our data set is relatively small. And then what we get is this AND data object. So this is just a data type in Python. It's like a matrix, and it's annotated with these observational and variable measures. So in this case, the observations are the samples, and then the variables are the genes. So we can see these are the properties of our and data object that's returned. And then to actually extract the statistics that we're interested in from this, those are going to be stored along the gene dimension, so in the variables. So we can call DDS, so that's our DSeq object dot var_m, so that's how we access all of the variable measures. And then I'm going to say I want to look at the log fold changes. So that's this attribute titled LFC. And then it'll print out as the log fold change estimates for each of the 21,000 genes that we included in the count matrix. Okay, so now I'll go over, before we actually get to the statistical testing of finding differentially expressed genes, I'm first just going to go over some visualizations that people will do to kind of just look at the quality of the data and sort of as a sanity check. So one of them that we talked about is the visualization of the spaces between samples through a dimensionality reduction plot. So we're going to do PCA here. The first step, we're just going to mean center our data, and then we'll call the PCA function, and I'm going to plot a three-dimensional PCA. So I'm going to extract the first three principal components. So we'll do that, put it into a data frame that we can then plot and color by the metadata. So in this case, I'm going to color my samples by the cell lines. And you can kind of see there's already in this three-dimensional space, we already see this separation between these two cell lines, so the purple and the red, along what looks like it is the second principal component, or first and second. But so this is one way you can sort of visualize, firstly, if you are going to expect differential expression across your condition of interest. And if we had other conditions in our metadata file here, we would also want to plot those to see if there are any potential confounding factors that might be driving the differences we see. So this is an important visualization step. And it can also be used to identify any potentially mislabeled or poor quality samples. So any samples that don't look like it belongs in a cluster, you might want to look into that sample and make sure that the data quality is good for those ones. Okay, so now for statistical testing, the DSeq stats class is going to be implemented from this the fitted DSeq dataset object that we had from before. So we're gonna instantiate this object, and we're gonna specify now. So here we have an alpha of 0.05. We can opt to filter out outliers with coexistence. If you don't want that, just turn it to false. And then this independent filtering step is what I mentioned before about DSeq doing its own filtering for genes with low counts. So since we already did that, I'm going to put that to false, but if you want a DSeq to do that for you, this would be the option that you use. So we're going to get our statistical testing results. And then we can print out the summary from the walled tests. So we're going to get this table with the base mean. And then, so this is the mean expression, and then the estimated log-fold change, along with the standard error estimate. This statistic is going to be that that walled statistic that I mentioned in the lecture, and then those are assumed to follow the normal distribution, and that's where we get these p-values. And then, so this table is going to contain all of our testing results. And to adjust those p-values for the Bonferroni, we can, or with one of the two methods we discussed last week, so we can choose to apply either Bonferroni or Benjamini-Hochberg adjustments here. We can do that ourselves. We can also take that from the table itself. So I think DSeq does the Benjamini-Hochberg method. But just to illustrate what it would look like for us to go and do this correction ourselves, I'm going to make a separate data frame first for storing those p-values. And then from the stats models library, I'm going to apply both of those methods. So the multiple tests has this method parameter. So this is the BH correction, and here we have the Bonferroni correction. So this makes it very easy to just feed in a list of p-values to the function, and then it'll give you back a set of corrected p-values. And so I'm adding those adjusted p-values to the table. So now I have a table with my log-fold change estimates, my unadjusted p-values, the average expression, and as well as my Benjamini-Hochberg p-values and my Bonferroni p-values. So this is what I'm going to use for our volcano and MA plots later. Okay, so just as a kind of a preliminary step before I would do those volcano and MA plots, I want to label my genes as whether or not they're significant, so whether or not they're upregulated, downregulated, or determined to be non-significant. So I'm just going to do that with some list comprehension. So it's a long line of code, but basically I'm just making new columns to mark my genes with their significance. So now I have those two columns here. And then this is how I will color my genes when I go in to do my MA plots and volcano plots. So here I have, I'm going to color any upregulated gene as red, downregulated as blue, and otherwise it'll be gray. So if you recall, the MA plot is going to plot that average expression on the x-axis with the log-fold change on the y-axis. So that's what we'll do here. And now we can see these up and down regulated genes. So yeah, I'm moving kind of quickly just for the sake of time. But maybe I think so this is the MA plot. You can look more closely through the code yourself later. For the log fold change plot, or for the volcano plot, let me see. Okay, so first just to limit the changes I have to make to the code, I am going to apply the LFC shrinkage that we talked about for DE-seq. And then regenerate the MA plot. So you can see that the shrinkage here and the effect it had on these genes with low counts. Okay, and now the volcano plot. So I'm going to just specify a LFC cutoff of 5. And then if you remember, the volcano plot is going to do the log fold change on the x-axis with the p-value on the y-axis. And so these are the points colored by or plotted by the Benjamini-Hochberg corrected p-values. Whereas if we were to do this with the Bonferroni correction, if you remember from last week, this is a more conservative correction. So we see that there's slightly fewer genes that are marked as significant when we use the Bonferroni correction method. Okay, so for the sake of time, let's see. I think just the main types of plots that you might want to make are covered here. So these remaining 2 methods, I may not walk through them step by step, but just in general for Lima, you'll be interfacing through R, RPy2 to run this method. And you can do very similar visualizations as we did with DESeq2. And then for the characteristic direction method, you'll have to do your own normalization before you can actually run the method. So the example I gave here is the quantile normalization, which I think you learned about in the normalization lecture. So we can see the effect of that library size normalization. And then the results for the characteristic direction method will be a table that looks something like this. So we have a coefficient that's computed for each gene and a significance level that's calculated. And so this is how you would rank your differentially expressed genes based on their contribution to the vector. And then all of these methods are going to differ slightly in the differentially expressed genes that they pick up on, but there should be some agreement. So we can kind of see the overlap of some of these methods of the differentially expressed genes that were identified from these 3 methods using this test data set.